cloud. And I will turn it back to David, right?
Or you could return it back to Dr. Pashton.
Okay, I'll give it to Tim. Here we go. Tim, you're going to become host shortly.
All right.
Did you share the screen also so that Tim can do any presentation?
Yes.
Yeah, he'll be able to do that.
That's part of being a host. All right. So first of all, thank you guys and everyone. It's nice to have this kind of interest in the general population about a topic as detailed as this. I can understand why everyone is interested, but it's great that you guys are willing to suffer through something I'm going to present to you for over 40 minutes. So I'll try to make it entertaining. If you have any questions or I'm using terms that you don't understand, stop me and say, hey, Tim, explain that, right? But I'm really going to try hard to get this at a decent level so that, I don't get stuck in technical terms. All right, so let me share my screen. All right, and you should see something that says coronavirus, et cetera, correct?
Yes.
Okay. All right, so I'm going to bring that up and let's start talking about it. So this is something that us in the professional community have been worried about for 20 years. I've had students email me after the pandemic started saying, I can remember when you talked about this in 2005 and warned that an epidemic was coming. The human population has gotten so large that we're starting to encroach into wild areas. We're starting to mix with animals that we didn't normally mix with before. And that is creating opportunities for viruses like this to jump from their wild hosts and into humans. And that's exactly what SARS-CoV-2 did. Now, this is not the first time this happened. This happened back in early 2000 when SARS-CoV-1 jumped and went into China and started killing people. Now, ironically, that virus was far more lethal with a 50% mortality rate. But because of that, its ability to spread was less. It would kill its host before that host could infect other people. But that was truly terrifying because of the mortality rate that the organism had. And ever since then, scientists have been concerned that something like this would happen. And in fact, it did. So, as you all know, this started at the Wuhan seafood market, is what was told in the press. I'm not going to address the ridiculousness of this starting in a Chinese lab. That hasn't been demonstrated at all, and that's just silliness. But it turns out it really didn't start at the seafood market either. There are cases going back all the way to November of 2019 of people coming into hospitals with infections that matched the SARS-CoV-2 virus. One of them then went to the seafood market and then probably started the first super spreader event. And the best stuff that I've seen has shown that that's the case. And the first case was diagnosed in December 1st of 2019, and then cases exploded in China and then spread worldwide. All right, so let's talk a little bit about the virus itself. This is a space-filling model of what the virus actually looks like. So this is a reasonably accurate representation of how coronaviruses are constructed. The virus is actually, the red part here is actually membrane, and it actually comes from the host cell. So this virus will bud out of the cell, just like a bubble coming out of an object. And it will have in it viral proteins. And the most important one is this big pink one. And it's called the spike glycoprotein, or S protein. You will have other proteins too. They're listed here. I won't go through them. They're important for the virus, but they're really not important for what we're talking about here. Okay, inside of that, you will have the nucleic acid, right? This is the genome, the set of instructions the virus has. Now, our cells also have sets of instructions. They're in our chromosomes. Those chromosomes are made out of DNA, right? And those chromosomes have, it's like a big library of recipes for how to make proteins. The virus is the same way. It will have its own little library here, which is much smaller because it's only about 30,000 base pairs. So it's about 30,000 units long, whereas the human genome is 3.2 billion units long. So it's much larger. But it's amazing what misery it can cause with just that little set of instructions. So you see that inside the virus right here, and it's coiled up. And this is a pretty reasonable representation of what the virus looks like. And they're called coronaviruses because it looks like a sun's corona or a solar corona surrounding the virus when you look on it in an electron micrograph, and hence that's how it got its name. One really interesting thing about SARS-CoV-2 is that its library, its genome, is RNA. It's not DNA. And that's something you only see in viruses. You don't see that in other organisms. Okay, so just a little bit about the genome. It's been completely sequenced. We can read the whole genome, look at it, and it's trivial to do this. I teach students how to sequence nucleic acids all the time. But it's a large genome for an RNA virus. It's about 30,000 what's called a base pair long, and that is much larger than your typical RNA virus. For example, the flu virus has a genome in total of about 10,000, so it's 3 times bigger, and it has its own RNA replicase. And this is something that's really important for understanding how to fight this guy later. So I'm going to go into a little bit of detail about it. This is the enzyme that copies its genome and makes new copies for when the virus replicates. The reason it has to code for it is because the host genome does not have this enzymatic function. We do not have replicases that will copy RNA into RNA. Another very important fact about this virus, which is very unusual for RNA viruses, is it has a proofreading function in its replicase. So if it makes a mistake, it can go back and fix it. Normally with RNA viruses, like the flu virus, that doesn't happen. Another RNA virus that I'm sure all of you have had is the cold virus. The cold virus is also an RNA virus. Its replicase doesn't have a proofreading function. Because of that, these viruses evolve very rapidly. That's why there's always a new flu strain coming out. That's why there's so many cold strains, and why you keep getting colds even though your immune system fights them off, right? So theoretically, this will not change as fast as other RNA viruses. Okay, you can see the different coding frames. The first one here codes for the replicase. Here's the spike protein. Here's the other proteins. Interesting to scientists, but probably not interesting for what we're going to talk about here. All right, so how does this guy replicate? Well, first of all, it binds to the ACE2 receptor. And this is a very interesting receptor, and this is different than other coronaviruses. And our ACE2 receptor defines where this guy can infect. And you have ACE2 receptors on your upper and lower respiratory cells that line the lungs and your respiratory tract, on your heart, on your kidney, and your gut. And that explains why this virus affects those areas. Okay, it enters the cells by binding this ACE receptor, and then being put into a bubble inside the cell, and then the RNA is released. All right, this RNA gets released into the cell, and it mimics, it looks like a recipe for making a protein that your cell can understand. So the normal way your cell makes proteins is it has this DNA library and it wants to do a recipe. So it makes a photocopy of it, we'll call it. And it makes this photocopy in RNA that comes out of the nucleus and it goes to a ribosome. And the ribosome synthesizes the protein, whatever the recipe calls for. Well, this virus takes advantage of that, and it looks just like a recipe that the cell would make for anything else, except it encodes viral proteins. When those viral proteins get made, they make all the different proteins that the virus makes. They also make instructions that cause the virus to assemble, make a bunch of copies of itself, and then leave the cell. All right. So the two really important things I want you to remember from this is that ACE2 receptor, defining where it goes, and that this guy comes in and is copied by the cell. It just takes over the cell and makes copies. All right. And there's more about replication, and this is like more details that if people want to know, I can go into, but I won't right now. Basically, it makes copies and then it buds from the cell. What it will do is it will affect cells first in the nasal cavity and the back of the throat. That's why you get sampled. They want to sample the back of your throat because that's where the first infections are. And it will start to replicate in those cells. The virus is released. It will then go to other cells in the same area, bind into them, get into them. And if your immune system doesn't control it, it'll eventually infect your lungs and cause COVID pneumonia. And that's when real problems start. Symptoms appear in 2 to 14 days after exposure. An early warning sign is a loss of smell, and you'll see that in about 2/3 of people. You can have fever in about 98% of people that are that are symptomatic. You'll have a cough in 76. About half, it will then move down in the lungs and cause shortness of breath. And in about half, you'll get fatigue and body aches. And I leave lots of links on here that you can go and look at articles that discuss all of this stuff in detail. The really interesting thing is a significant number of cases of SARS-CoV-2 are asymptomatic. It just replicates in your nose and in the back of your throat, doesn't really give you anything more than you'd think is a cold, and yet you're spreading virus to everyone else. This is a big difference between this virus and SARS-CoV-1. SARS-CoV-1 always pretty much moved to symptomatic cases. You never saw this asymptomatic spread so much with that virus. And that's one of the reasons SARS was able to spread around the world pretty quickly. So what happens? The majority of cases are mild. About 40% will be completely asymptomatic. Another 40% will have very mild symptoms. However, severe cases on the other 20% happen that need hospitalization and management. Severe cases, pre-existing conditions make it more likely. Those with heart disease, about 10.5% will go to severe cases. About 7.3% of diabetics will go. Chronic respiratory disease, 6.3%, high blood pressure, cancer. Only about 0.9% of those with no pre-existing conditions will progress into serious disease. However, it's not zero, right? Hospitalization and death is possible in anyone over the age of 19. And in fact, now that we have more data, this is data of March 16th of 2020. So this is quite a while ago. And while they didn't see cases in younger people, as the Delta variant has come up, you're seeing more infections in younger people. And it's honestly more of the population is getting infected. So you're seeing a few cases. But as you can see, the likelihood of significant hospitalization, ventilation, that's what the dark, lighter, like light blue, medium blue band is, having to be ventilated, etc., and death increases as you get older. All right, so it becomes more and more serious disease as you get older. So how do we treat this disease? Well, there were a number of potential targets that could have been attacked. One of them was endosome fusion. A big thing that was looked at early was chloroquine or hydroxychloroquine. This turned out to be largely unsuccessful. As a scientist, it was quite unfortunate. This became a political football because early studies indicated that hydroxychloroquine could maybe have a positive effect in treatment. It did work in the test tube, but clinical trials showed that there was really good rigorous trials showed that it was not successful. And that what the hydroxychloroquine and chloroquine did is they prevented the virus entry, this fusion and entrance into the cell. Other drugs, such as remdesivir, attack the RNA-dependent replicase. And that's why I spent time on this, because this is a great target. This is something that the virus is absolutely dependent upon, but we don't have this function. So any drugs made to inhibit it should not affect humans. So it's a great target to go after. Remdesivir has been approved for emergency authorization use, but I'll be honest with you, I've looked at the trials for it, and while it does show some effect, large studies have shown that it's no more effective than hydroxychloroquine. So my question is, why are we prescribing remdesivir and we're not prescribing hydroxychloroquine? There isn't any, doesn't seem to be much of a benefit.
I don't think that's a fair assessment of remdesivir. I think it does have, but only in selected patients.
Yeah, I think a lot of the time, well, I'm going by the largest WHO studies, and you can go look at that reference. WHO doesn't recommend remdesivir very much. But you can go look at it. My feeling is it doesn't give a strong effect. But honestly, right now there wasn't good treatments, so I can see why people used it, but it really doesn't do it, especially in late cases, it doesn't do much effect. If you use it earlier, it might have a decent effect, but I just, again, have not seen good data yet to support the use of remdesivir. I mean, I'm being probably pretty harsh on it, but I mean, that's the way, from my read of the trials. And again, absolutely go read that reference and make up your own mind. Another successful treatment is monoclonal antibodies, and these have shown a positive effect if used early enough. And I think this is a reasonably effective treatment. Steroids have also been shown to mitigate the immune response, such as dexamethasone and prednisone, and they decrease the risk of ventilation and death in patients. Again, if used timed at the right time. Unfortunately, some individuals wait too long to go to the hospital, and by the time they get there, it's too late to use these kind of treatments. Okay, so that's where it stands right now as far as treatment for this disease. Except for there's a new drug, Molnupiravir. I think that's how it's pronounced, Molnupiravir. And this is kind of exciting because this is a drug that was developed to go after RNA-dependent RNA polymerases. It was originally developed for flu. And if you look at the drug, it's right here on the bottom. And if you look at its structure, it is what's called a nucleotide analog. So it looks very similar to cytosine, and the replicase will actually mistakenly put it in. And once it gets put in, it will incorrectly pair with bases and cause all sorts of mutations in the RNA. And so it's a great inhibitor of, it caused all sorts of problems and caused viral death. It's worked very well in the test tube. One of the really nice benefits of this is the proofreading capability of the replicase does not recognize this as an error. So it won't go back and fix it. And that's really important. So it can get past that. So again, this is a great enzyme to attack, because it's essential to the function of the virus, and we don't have an enzyme that does this. All right, so there was a recent drug trial. They looked at, and again, here's the reference to an announcement on the information that was released. You can look at that. They looked at 775 patients that had at least one risk factor for a poor outcome. So this was a significant group that was at risk, and it reduced the risk of hospitalization by 50% in the test group versus placebo. This is an excellent study. It was reasonably large. It was double blind, meaning the physicians and the students and the patients did not know if they were getting the drug or not. And there were no deaths in the test group. There were 8 deaths in the placebo group. The results of the study were good enough that the review board asked for a halt to the trial and for it to move forward and not continue it. And that is like the best outcome you can get in these kind of drug trials. Also, there were very few side effects of the drugs. And in fact, any kind of side effects that were reported equally in the test group versus the placebo group, saying that it probably wasn't from the drug itself. I think this is going to be a very important drug. We'll see going forward, but right now the results look very promising, and they're quite a bit better than anything that's gone forward yet. Another nice thing about this, Remdesivir and the monoclonal antibody treatments have to be given intravenously. Molnupiravir can be given orally, so it's a much easier drug to administer. So I'm pretty excited about the results for this one. All right, so the other effective treatment are the vaccines. One thing that I was always pretty confident, you can never know on these vaccine trials, but the people that I spoke to that really know this and develop vaccines said that this is a pretty easy vaccine target. Everyone knows that coronaviruses, they've been studied for years, and it's known that your body raises a strong immune response against the S protein, that spike protein on the surface, and that's what gives you immunity. So we had the sequence early. We were able to make vaccines pretty quickly, and the people in the scientific community, for the most part, were optimistic that we'd be able to create some pretty effective vaccines. Another nice thing about this is that the spike protein has limitations. It can't mutate massively. Now, of course, it's going to mutate, but it's a good target. It's not going to change, massively over time. Hopefully, we'll see. Now, 3 vaccines that are approved for use in the US are the Pfizer BioNTech vaccine. The first trials had a 95% efficacy in the phase 2-3 trials. There were 100 cases of infection in these trials. They had 43,661 participants. And the way they do these trials is obviously you can't vaccinate people and then challenge them with the virus. What you do is vaccinate people and then follow the whole cohort and see who gets exposed naturally and who gets sick. In those that were fully vaccinated, there were 100, actually, that's not correct. In the trial, there were 170 cases of infection, 162 in the control group, and only 8 in the test group. All but 1 severe case was in the control group. There are very few side effects, although there are more. I don't think these numbers are, I haven't looked at numbers for like generally, but you'll have fatigue and headache, etc. But it's an extremely effective vaccine at protection. It had a 95% efficacy. The Moderna vaccine was just as good, had a 95% efficacy also in the phase 2 trial with 30,000 participants, 95 cases, 90 in the control group, 5 in the test group. Again, all severe cases were in the control group, so it was very protective. The Johnson & Johnson, both of these are mRNA vaccines, and the way they work is they use a lipid delivery vehicle to deliver the RNA directly into the cell, and then your cell ribosome is tricked into making the spike protein. It displays it just like it would as a virus, but of course you're not infected. But it sure looks like your cells are infected, and then you generate an immune response. That's both how the Pfizer and Moderna vaccines work. The Johnson & Johnson vaccine uses an adenovirus, and this is a delivery vehicle that delivers a piece of viral DNA encoding the spike protein into the nucleus of cells. The DNA delivered into the nucleus then makes mRNA, then the spike protein is synthesized and the same thing happens. Now, very important, this viral DNA from the spike protein is not inserted to the host DNA, right? So that was something that kind of went around, and it's not true at all. Okay, so what can you do? Obviously get vaccinated. One thing that I've been following that I found really interesting, if you look at cases of COVID in Dane County, Wisconsin, that has vaccination rates of 12 and over about 85%. Okay, so I'm gonna move this here. About 85%. Cases per 100,000 is 26. Okay, and we have a lot of college students. We are at a much higher concentration in this county than other counties in Wisconsin. And you'll notice if you compare our November, December peak last year to what we're experiencing now with the Delta variant, you're not seeing as many cases, right? It's about, I don't know, 5-fold less, right? Compare that to Marquette County, Wisconsin, which has a vaccination rate of 12 and up of 55% cases per 100,000. You have 115, and they are progressing to the point that they may even surpass the number of infections that they had back in last year. So the vaccine does make a significant difference, right? So the best thing that you can do right now is get vaccinated. There are other things that you can do to keep your immune system healthy. They're going here. Get enough sleep, eat fruits and veggies, don't smoke, drink moderately if at all, try to minimize stress, right? And then, of course, try to fight misinformation, which is incredibly difficult in these times. Okay, that's pretty much my presentation. In looking at this, I'd be happy to take questions about anything to do with this. I'll try to answer the best I can, but I will honestly tell you if I don't know.
I have
Yes,
one.
yeah.
We hear a lot in the news about B cells and T cells. Is this something that we need to be concerned about? What is
Well, okay, so B cells and T cells are things in your immune system. You hear a lot of talk about sterilizing antibodies and how they're important to an infection, and that they will measure that to measure the quality of your immune response or how well it's working. And that's not the whole story. When you get infected with something or you get a vaccine, you will raise both a B cell response, which makes antibodies, and you will raise a T cell response. In the case of SARS, the T cell response is targeted toward getting rid of infected cells. So those T cells will go out, and once they're activated, they'll be able to identify cells that are infected, and they will be able to kill them, right? So both responses are really important. When you're immunized or when you're infected, you build up memory cells, memory B cells and memory T cells that spread throughout your body, park in your lymph nodes, and they're like your reserves. They sit and they will wait for the next time an infection occurs. So while it is true, it's better if you can maintain higher antibody titers. If those antibody titers go down, you might be able to get infected, but that doesn't mean you're naive. You still have those memory B and T cells, and those can be reactivated, and they can absolutely help you fight the infection and hopefully do a better job with it.
Okay, well, that clears it up. The B cells are related to antibodies.
Yeah,
and
the B cells
are related
change
to
into
removing
plasma
infection.
cells that make antibodies. Yes.
And the T cells actually deal with fighting and removing the infection.
Well, they'll fight and remove cells that are infected. Yes.
One thing I didn't know is that the membrane of the virus that we see on TV all the time is a membrane of the actual cell in our body to begin with that we're
Yes.
infected with.
Yes.
That's never been shown on TV. It always looks like that the virus is that round thing with the spikes poking out.
Yes.
The spikes are coming as a result of the virus that's inside the cell.
Yeah. So the virus, what happens is that virus gets made into all the various proteins, and each of them has instructions on that protein that tell the cell where to put it. And the spike protein and the E and M protein all go to the membrane, and then they have the instructions to cause them to congregate in an area. And then you bud out of that area. And as you bud that membrane out, you have all those viral proteins on it. And that makes the virus or viroid.
People who have had the virus and survived, what are the thoughts about having the shot, and how effective is protection just having had the virus itself?
Okay, so this is speculation because we don't know. SARS is so new, right? But with past coronaviruses, that protection wanes with time because of proteins in the virus that actually short circuit parts of your immune system.
Actually, there actually are publications that show that just from being infected is not nearly as good protection as from the inoculation plus the infection.
Yes, absolutely agree. And the reason that is because, and again, this is speculation on my part, is that you're only getting exposed to the S protein when you're vaccinated. So you'll raise this huge immune response against it. You're not getting exposed to the proteins that the virus has that kind of weaken your response. And so a viral infection doesn't raise the kind of antibodies. Okay, again, that's speculation on my part. Part of the other thing is you just getting hit with another set of antigens that jack up your immune system. But yeah, absolutely, vaccination is better than being infected by the virus. And even if you've been infected, a vaccination will increase your immune response to a higher level and be more protective.
Thank you.
Question for you. There's a lot of information out there concerning the immunodepressed people, those that have cancer, diabetes.
Mm-hmm.
Should they or should they not get the vaccine?
Yeah, well, the best thing that we can do for them is vaccinate everybody else and stop decrease the circulation of the virus in the community. It's worth a try. It's not going to hurt them, but there's a concern that they won't raise a vigorous immune response. No, but as far as is the vaccine more dangerous for them? No, I've not seen any data that shows that it is.
I have a question. Peru seems to have almost twice as many deaths per capita of any country or state in the world. Is there any explanation for that?
I've heard that statistic, but I don't want to speculate because I haven't read anything on what's going on there. I think, yeah, I honestly, I don't want to say because I haven't.
One possibility is it has to do with reporting real infections and how accurate the statistics actually are.
Yeah.
Another question would be about sickle cell trait. Has that proved to be a pre-existing condition?
Not that I'm aware of.
No, I don't think so. Obesity is a,
Yeah.
is a,
Obviously, sickle cell anemia would be a big pre-existing condition, but and you can die suddenly just from having the trait. Sometimes if you're oxygen short, heavy
Mhm.
exercise,
Mhm.
would make sense that COVID might drive you into that lack of oxygen.
Yeah, but I would think that that would have come up in the data. I mean, as a
I haven't-
comorbidity.
I haven't
and
seen
as-
anything to.
Yeah, I haven't seen anything to support that
that.
at all.
Yeah, this is Doug Johnson hidden behind a black screen. Apparently, my camera is not on. There's been some discussion among acquaintances of mine about getting your, oh, no, I forgot the word I wanted to know. Your getting tested to see what your antibodies are.
Mhm.
Some people are saying, oh yeah, you should really do that, and others are poo-pooing it. What's your thinking on those?
I don't
Okay,
think-
so if you're someone who's had the illness and you're trying to justify not getting vaccinated, no, go get vaccinated. If you're just curious, I don't know. That's kind of wasting resources, in my opinion. Just assume that you're probably okay. I haven't seen any. I mean, normally, if you have higher antibody titers, theoretically, you should be more resistant to infection. I can understand why people want that for peace of mind, but there's not a real good understanding yet of what the floor is. How low can your antibodies go and you're still protected? And again, as Jeff had asked the question of B and T cells, even if your antibody levels go
Right.
to an undetectable level, that doesn't mean that you're not protected against the virus. We don't know that yet. So would I ever go get tested? No, I wouldn't.
I don't
just
think there's any useful way outside of a controlled study to interpret results that you would get from a test like that, unless you have
Yeah.
a specific question that you're asking, like, did you ever have an infection? That's when an antibody will answer the question for you. But using it to interpret risk, I don't think is developed in a way that's useful.
Yeah, it's not. No, there are other things like getting tested for chicken pox. If you've ever had chicken pox before, they understand that much better, and that they can tell you what that means by your antibody titers. But yeah, totally agree with what Philip said.
Thank you.
You bet.
Regarding the Pfizer, how important is the third shot, the booster?
For people over 65, I would highly recommend it. I think it's important. There is evidence that shows the efficacy goes down, especially with the Delta variant. Unless you had trouble with the previous shots, and I mean like an allergic reaction or something, then it's not recommended. But it is still controversial. There's a significant number of scientists that think it's unnecessary because the efficacy that's really important in their mind is hospitalization and death. Those numbers haven't gone down. It's still almost just as effective against the Delta variant as it was against the Alpha variant and earlier variants. So, but I think what they're trying to look at is preventing further spread. But if they really want to do that, they got to get the people that are unvaccinated vaccinated, right? On the other hand, if you're an individual that's in a high risk occupation, like you spend a lot of time with college students or large groups of people, all right, I would recommend a booster in that case.
Or you go to football games.
Oh yeah, or if you want to do those things, absolutely. Because football games, the rest of Wisconsin's also coming into this area. And unfortunately, a lot of people in outlying areas are not making what I would consider good choices. if you look at Wisconsin, it's bright red with infection, except for this little island called Dane County. We're doing really well here. So
If you get
Yeah.
the booster shot, I think the chances of having a reaction to it are higher if you've had an if you've got immunity because of the earlier shots. Is that correct?
that is correct. Your chances of side effects increase. I will tell you, I just got the booster last Friday. I was not feeling very well on Saturday, but it was like body aches and it felt like a very mild flu. And,
Well, in that
and it was
case,
gone within 15, 20 hours. I'm willing to go through that.
the side effects then indicate that you had immunity and you're getting more of it. It's a good thing.
Maybe. I'm being super careful here, Jeff. I think you're correct, but I've never seen any good data where someone's done a study looking at the amount of side effects that someone had and then measured their immunity. That would be very interesting.
Yeah, I haven't seen anything on that. I am reassured that I have some side effects when I get the vaccine, and I've
Yeah.
gotten the booster, and I had just what you described for about a day. I felt kind of off and tired, and
Yeah. And I think Phil and Jeff, you're right. I think that does mean that there's a strong immune reaction. That's what's happening. I mean, those are cytokines going through your body, reacting to what you were given. And
Right.
so that does seem, it would make sense that means that you're then raising antibodies, you're going through an immune response. But I'd like to see data on it. I'd like to see that measured.
Well, meanwhile, I'll look at it as a positive effect. It means good.
Yeah. Yeah.
Yeah, I think I'm going to do the opposite because I had, other than a little bit of a tenderness at the site of the injection, I had no residual effects. And both of my children and all of our grandchildren were pretty much laid low for a day.
Mm-hmm.
So I'm hoping that they're right when they say, nah, you can't really tell from that.
Yeah, I'll tell you this, and I'm pretty confident in this. I think that having a reaction does mean an immune response, but not having a reaction doesn't mean that the flip side of that, not having a strong reaction against the shot does not mean that you don't raise protective immunity. Again, that's not been studied, and those are two different questions. So I think you're fine in assuming that it is protective for you.
Thank you. I'm going to tell everybody.
Is there any significance to the type of RNA virus? I'm looking at it, looks like it's single-strand positive.
Yes. So yeah, that's just the way the virus replicates. Is there any significance to it as far as no. I think a single-stranded RNA virus, positive or negative, is going to have about the same mutation rate. They tend to be higher, but like I said, this one isn't as high. If we could just get the number of people getting infected and spreading the virus down, if we could drive down the numbers of infections, we could stop the generation of variants and we could stop this cycle. But it's a matter of getting that down. Now, I'm optimistic, maybe incorrectly so, that a combination of herd immunity and the vaccine will get us out of this wave. Hopefully we'll have a calmer winter than we did last year. And then with the new treatments coming next year, that'll get us a way out of this, hopefully permanently. I really do think some of the drugs coming up, and the one that I talked about is just one example, right? Molnupiravir is just one example. There's a number of other drugs in the pipeline that haven't gotten as far, but they're also looking promising. There's also a nasal vaccine that's coming. And that actually has demonstrated in mice a sterilizing immunity. that seems to be robust. And it's because of how it's delivered. It's delivered the same way the virus is. It's delivered into the nose, and it replicates in those cells instead of in your arm, which then spreads throughout the body. That's why I think you're seeing this. You're seeing a systemic immunity, like, because if you get hospitalized, you have a systemic spread of the virus. If you come close to death, you obviously have a systemic spread. So then the whole immune system's involved. With infection, with a lot of people, it's just in the upper respiratory tract. If we can get the immune response there, hopefully that will get rid of, will provide a better immunity. And so far, but this is just in mice. So, but it's going into human trials now. Hopefully that'll work.
Another question, there are lots of big countries who have had very few deaths. China, India is pretty low, Indonesia, Pakistan.
I wouldn't trust the data on India if I were you.
Okay.
Yeah, I mean, they have political issues. They have their health system isn't as robust as ours is. So I wouldn't use the data on India.
The data I've seen suggests that India has a very high number of deaths, and Brazil, for example, has a huge number of deaths. And what was the other country you mentioned?
China.
China's had a lot of deaths, but they've also been, very heavy-handed about enforcing public health measures once they realized what was going on.
Yeah. And I think that might, I don't think they've had as many infections as we've had. And that's why, because they've been very heavy-handed in their management of it. Everybody in China is required to carry an app. If you go to the mall, you have to check in a QR code that says you were there. They monitor every single human being in their country and everywhere you go. That's not going to work in America. You'd never get Americans to cooperate with that. We can't even get people to take the vaccine.
So these countries that
One country that has been doing well is Australia. My wife's Australian. She corresponds with her sister frequently, and they are going about life as normal in Australia. But the government has a very strict policy. If anybody is infected, that they quarantine everybody, that they do a contact trace because they know where people have been. And it's a small country, and they've limited travel, so you can't move very frequently from one city to the other.
And they can't just drive in with their car.
No, it's the only continent that's all by itself. But the limited population, everybody's along the coast.
I mean, New Zealand has also been very successful.
Yeah, but New Zealand's a lot smaller than America.
Right.
Yeah.
Are these countries that have been successful, will they be successful in the long run? I think of Singapore, which had fantastic control until it turned out their foreign workers ran through them.
Yeah. So infection control can flatten the curve of infection and slow it down and give you time to make mitigating efforts. So we have those now. If they can get their populace vaccinated and the vaccines remain effective, absolutely they can keep the number of deaths down. But in the case where you don't have a mitigating effort, let's say we weren't able to develop a vaccine, eventually everybody would have got it. And but again, that's not how the world works. We have technology, we have scientists, we have physicians that just can do a phenomenal job. I'm amazed at how quickly this vaccine was developed. And as long as the governments continue with that, they'll be fine. So yeah, I expect that their deaths will never reach the levels.
But you have to get people to buy into it. I mean, we've had 700,000 people die from this. You have to, I mean, for reasons that could be a completely separate kind of discussion. It's like we have a war and we mobilize an army of physicians and vaccines and medicines And then 40% of the population basically deserts because they refuse to get the vaccine. They're just like deserters in a war.
Now, that's a great analogy.
Except they don't even have the, I mean, at least in a war, you have good reason to desert because people are shooting at you. They don't even have that excuse.
Yeah.
So,
Yeah. Yeah. It's the power of propaganda. The internet's a wonderful thing in some ways, but it's terrible in others. You can get so much garbage. I blame Facebook and Fox News and these other sites that they're pushing all this garbage.
well, they've turned a public health issue into a political issue, which
is just ridiculous.
Doesn't make any sense.
Yeah, it's unfortunate.
This is Phil Cox. I have a quick question about my situation. I'm 81 years old. I've had both vaccines, the first and second shot, Pfizer. I'm scheduled for my booster tomorrow. I have, like I said, I am 81. I do take 14 different medications during each day, which is a lot. I got a little bit of this and a little bit of that, but nothing that's got me yet. I assume with that situation, I should go ahead and I didn't have a reaction of any kind except just a sore arm for a little while. The previous 2 shots, there's no reason why I shouldn't go ahead and get the booster, right?
Okay, I'm not a physician, Phil. All right.
Okay,
What's your doctor say?
I am a physician, so I'll
Yeah,
say something about it.
please do.
The simple answer is probably you should, but you should talk to your doctor based on what the other things are, because maybe some of the medicines you're taking I don't know. I mean, it's always bad to give somebody medical advice when you don't know the whole story, and I don't know the whole story. I'm 81 years old, and I don't really have any active issues, and I got the booster without much problem. But you need to ask your physician whether there's any reason why you shouldn't get it.
Well, I had no reaction at all to basically to the first 2 shots, except arm was a little sore for a couple hours, and that was it.
Philip, I think Peter is saying that it depends upon the medications you actually are taking. Not knowing what they are, your physician might know
Right.
if some are okay and some aren't.
Yeah. Okay.
I mean, you've already had the first 2.
Right.
That,
Right.
I would be very surprised if there's any reason why you couldn't get the booster, but you should talk it over with your physician.
Okay. Thanks.
Your comment a little while ago about the nasal vaccine struck a chord with me, because recently I heard about in development is a patch that would deliver the vaccine to your skin instead of to your muscle with several tens of times better effect. Have you heard about this, and what do you think about that?
I've not heard anything about it. So yeah, I can't comment.
I've seen mentions of a patch, but I haven't seen anything to suggest that it has better penetration than other than maybe because the rate of administration would be slower with a patch. It would go in a little bit at a time. Maybe that's better. I don't know. I haven't seen anything to say that it would be better, and I haven't seen anything to say that it's ready for prime time.
No, what I saw or read was just that it's in development, and the theory, I guess I should call it, is that there's a lot more blood vessels going through your skin, which is why it would be more effective.
Yes,
At least that's the way I remember it.
yes, I think that's right. That was the justification I saw for doing it.
Very good. Let's see here.
I mean, I think that if this drug Molnupiravir turns out to work, it would go a long way toward being able to control the epidemic because it can be taken orally. It can prevent people from being in the hospital. And in theory, it could even be given to people who are exposed but haven't been infected yet. But it remains to be seen, number one, whether given to a huge number of people, there will be some unexpected side effects.
Yeah.
And number two, given as an individual drug, whether the virus will develop resistance to it.
That's my main concern.
Whether it needs to be given with a second drug like we did in HIV.
Yes, yes, that's my main concern. Now, one mitigating factor is going to be that this RNA virus has that proofreading capability. So it's not going to mutate as rapidly.
Right.
But I mean, this is when you put this kind of selective pressure on any organ, you know, anything, any enzyme, I mean, it's gonna probably at some point escape it, you know. And so, yeah, my main concern is that we start using blanket prescriptions of this instead of, you know, using it selectively. And the articles that I've read, the experts are talking about exactly what you said, Philip, is using this with other drugs and hopefully, you know, making it less likely that you generate resistance.
I just got one comment for both Tim and the doctor, since there's two Phillips on the line. 6 months after I got my second shot, I did develop that rash they talk about. And been in contact with the National Institute of Health through their virus effects reporting system and V-Safe that and I did get the booster, and so they're making sure that I monitor it to see if I have the same effect.
So when did you get the rash?
It was about 6 months or 5 months after the second shot.
Yeah, I'd be surprised that an adverse event would take that long to manifest. I don't know, but you know, that could be anything too, you know.
Yeah, well, what they say is it would take a lot more research and studies to I sent them the pictures and everything, and my doctor had her opinion of it, and said it may or may not be. And
was it a generalized rash all over your body, your chest, and or just in one place?
Generalized, is blistering in some areas, changed to hard pimples. And what was interesting, they prescribed a steroid patch. And then I started taking Hydroxychoroquine, whatever that drug is for arthritis, and cleared it up, which I also found interesting. But now after I took the third shot, they're pretty much having me monitor it just to see if it happens again.
I would be surprised if the rash was related to the either an infection with the virus or a vaccination, if it only occurred 6 months later. There are viruses that do things like that, but like herpes viruses, but those viruses integrate themselves into some cellular component, and or Epstein-Barr, for example, we carry those around for the rest of our lives, and they act up from time to time. But I've not seen any evidence that coronaviruses do that.
I don't think it can. It's an RNA virus.
Yeah.
Now, both the other two, Epstein-Barr and the herpes virus, are both DNA viruses that make episodes that go into the nucleus and hang out. So.
Yeah, what is it, VAERS or whatever for the virus reporting system sent me pictures and said, is this what it looks like? And so I sent them pictures back, and it was very similar. So evidently there is something along that line, but I can't say anymore because I didn't study it.
I've never seen reports of such a thing, but remember, a rash, your body has only certain ways it can react, and your immune system has only certain ways it can react. And a rash is a reaction pattern. And it can be triggered by many, many different things, including drugs, foods, and infections, or certain kinds of unusual systemic illnesses. And so I would, if I had to guess, I'd guess that there's some other explanation for the rash. But on the other hand, strange things happen. And my opinion would be that there's no reason why that should keep you from getting a booster shot.
I already got the booster, so I'm. They want me just, yeah, they want me to keep an eye on my skin and see if it happens again. And then they want to do some more studies. But
Right.
evidently they did send me some pictures, so there must be other people that are having the same effect. And it's a interesting pimple that they have
You
pictures
can
of.
find lots of pictures of generalized rashes on the web. And you'll see lots of them from ampicillin or any number of conditions. So I'm not saying that they're not related. I can't say they aren't, but I would be very surprised if they are.
Yeah, well, I was very hesitant to say that the vaccine caused it until they actually sent me the pictures that said this is a rash that's been reported.
Yeah, I agree with Phil. I'm really skeptical that it is related because the time lag is so long. 6 months later, if it was a week later, okay, or 2 weeks later, yeah, sure. But 6 months, there's a million different things that could cause a rash.
Well, I'm going to look into it.
There you go.
Well, thank you. Thank you, Dr. Poshan.
I'm happy to help. It was a very interesting discussion. It was great to
Yeah.
answer questions and hear everybody's
Thank
thoughts.
you.
Yeah. And before everybody else leaves, we're going to have our annual elections, which we missed last year.
Okay, I will see you guys later. Have a good evening.
Thank you.
Okay, so no, I'm just going to leave.
Wait
the
a second,
meeting,
Tim. You can't.
and I
you
will
can't
give it to
leave.
Jeff. You are now the head honcho.
Oh, am I in charge?
You're in
Okay,
charge. Bye-bye, guys.
Okay, thank you, Tim.
Hey, Tim, we're going to vote you in for something.
No, that's okay.
Okay, thank you. Okay, I'm the host, but that doesn't mean I'm in charge of the election.
Okay.
So
I'm going to.
I'm going to stop the recording now.
Yeah.
Okay, I'm stopping the recording.